we just looked at how an action potential can travel down an axon. Now let's take a look at some of the factors that can affect the speed at which that happens. So we have two main factors that are going to affect the conduction speed of an action potential down an axon. The first is axon diameter. So what you're looking at here is a cross section through a nerve. Okay, a nerve is a bundle of axons. You're looking at a cross section through a nerve from the squid. And what you can see here is you have this one giant axon, the squid giant axon. It's about 0.8 millimeters in diameter, okay? And a bunch of other small axons, okay? And the point that I want to make here is that this axon, the squid giant axon, is going to conduct a lot faster than these other little axons right here. And the reason for that is that larger axons, axons with larger diameters, are going to conduct action potentials faster than axons with smaller diameters. The reason for that is that with a larger diameter axon, you get less resistance to ion flow down the axon. So the ions that are coming in and depolarizing that next part of the membrane, they're going to be able to move more easily down a big fat axon than a little skinny axon. So you can kind of think of that as being like a water pipe, right? So the wider a water pipe is, the easier it is for water to move down it. The skinnier it is, that water's gonna have a harder time going through. However, we can't just have big axons everywhere, right? Because they're big, they take up a lot of space. If we wanted to be smart and we did that just by having big fat axons, we would have to be gigantic, okay? So there's another solution, okay? Another factor that affects conduction speed of action potentials down an axon is myelination, okay? So just to remind you what myelination is, I want to remind you that some axons have this myelin sheath on them. The myelin sheath is formed by oligodendrocytes or Schwann cells wrapping around the axon. Okay, so we have these segments of myelin sheath, and in between those segments of myelin sheath, we have these bare areas of membrane called nodes of Ranvier. So bare meaning not myelinated, okay? Okay, so looking at this picture here, what you're looking at on the right is another cross section through a nerve. Each of these little kind of grainy things is a cross section through one axon, okay? And all of these little axons, each one of these little axons can conduct action potentials just as quickly as this big squid giant axon right here. How does it do it? Myelination. Okay, so myelination is really a huge factor in speeding up action potential conduction. So let's take a look at how that works. Okay, so first we're going to look at what happens in an unmyelinated axon. When we conduct an action potential down an unmyelinated axon, that's going to look just basically the same way that I described it in the earlier video, where you have sodium coming in, depolarizing the next section of membrane. That's going to open sodium channels. That's going to allow more sodium to come in. That's going to depolarize the next section of membrane, and so on, okay? That takes time, okay? Another thing that happens in axons is that as the action potential travels down, if we don't have ion channels here, if we don't have sodium channels here, as the action potential travels down, some of those ions are going to leak out of the axon, and that's going to make that action potential get weaker. So earlier when I said that an action potential stays the same size. That's not quite true. It degrades a little bit until it makes it to the next place where that signal gets regenerated. Okay, so that's what happens in an unmyelinated axon. If you look at a myelinated axon in comparison, what you have is, first of all, let me just describe what's going on here. So you have in the nodes of Ranvier, you have a high concentration of sodium channels, voltage-gated sodium channels, okay? And then in the myelinated areas, you have no voltage-gated sodium channels, okay? So when that sodium comes in and depolarizes the membrane, we generate an action potential, or we regenerate the action potential that came from earlier up on the axon. That action potential travels down the myelinated part of the axon. It doesn't degrade, or it degrades very little, because there's very little current leak, very little ion leak out of the sides of the axon here, because you just have this big thing surrounding it. Okay, it's like this insulation that's surrounding the axon. So that signal stays strong, and it can stay strong all the way till it gets to the next node of Ranvier. Okay, once it gets to the next node of Ranvier, we have a lot of sodium channels there. We're going to regenerate the action potential there. Okay, so signal stays strong. We don't have to open channels at each step along the membrane. Instead, we can just travel right past that myelin and get over here to the next node of Ranvier. And in that way, that speeds up action potential conduction, because we're not taking the time to open up all these sodium channels right here. So it's just going from here, zipping on over, and then it's going here to get regenerated and zipping on over to the next node of Ranvier, okay? This type of conduction we refer to as saltatory conduction, because the signal is jumping from node to node. So those of you who know Spanish, saltar means to jump, right? It's probably similar in similar languages, but I don't know those. So yeah, saltatory conduction. In a myelinated axon, the action potential basically jumps from node to node, okay? All right, so this is just a reminder for you and for me to tell you to watch this video. Okay, there's a very nice video that I will be posting that shows the propagation of an action potential, both in unmyelinated and myelinated axons. It has a really nice animation that really gives you a good sense of what saltatory conduction looks like. So I urge you to watch that. And then finally, this is your study guide, right? So here are your learning goals for this part of the lecture. Here's your vocabulary. See you next time.